course, it's a common observation that offspring resemble their parents. Humans have baby humans, they don't have baby cats or dogs or lions or tigers. But as well as that, children resemble their parents more than they do other members of the same species. It's usually possible to detect family resemblances. And the reason for this is that these inherited characteristics are determined by inherited genetic material, the genetic instructions. And we get half of these from our mother and half of them from our father. At least that's true in terms of the nuclear DNA. Half is from the mother, half is from the father. The mitochondrial DNA, actually all of that comes from the mother. But we are a combination of our parents' genetics, pretty well half and half from your mum and half from your dad, half from each parent. And I guess what genetics is the study of how this inheritance takes place, the nature of the genetic material and how this genetic material is passed on and how this manifests itself in individuals, the study of inheritance. Now, etiology refers to that which causes disease. So the study of etiology is the study of the causation of disease. Now in the UK we spell it with an A. If you're reading texts from the United States, they spell it with an E, they leave the A off. So whether you use the term etiology or etiology, it means exactly the same thing. It's that which causes disease. And we need to understand what causes disease, because if we understand what causes disease, we can then take steps to prevent the disease from happening in the first place. So really all our health education and health promotion activities are based on our understanding of etiology, our understanding of how a physiological process is changed into an abnormal pathophysiological process, the study of the cause of disease. Now classically, etiology is divided into endogenous and exogenous etiological factors. An exogenous etiological factor is from outside of the organism, the environment affecting the individual. An endogenous etiological factor is one that arises from within the individual. And really the only true factors that arise from within the individual completely are genetic factors. So genetic factors arise from the individual and have the potential to cause disease. Exogenous and endogenous etiological factors. And we're going to start off by looking at the endogenous etiological factors, that is the genetic factors in the causation of disease. Well, healthy humans all have 46 chromosomes. They're located in the nucleus of the cell. And the term chromosome actually means coloured body. Chromo means to do with colour. Healthy humans have 46. They inherit 23 from their mother and 23 from their father. And it's the chromosomes that carry the DNA, the deoxyribonucleic acid, the genetic material. Now, chromosomes are also made of structural proteins called histone proteins. So the chromosome itself is made up of a combination of structural protein and the DNA, the deoxyribonucleic acid, which carries the genetic material. And all mature cells have 46 chromosomes, the only exception being mature red blood cells. Now, the red blood cells had chromosomes when they were being formed in the bone marrow, but in the circulation they've actually lost the chromosomes because they're no longer nucleated cells. But other than that, the chromosomes are present in all cells in the body. What I've done here is I've taken the previous photograph and cut out the individual chromosomes and arrange them into pairs called homologous pairs. So on the top left we see homologous pair number 1, then 2, then 3, then 4, then 5, and then 6 on the top row. These are homologous pairs of chromosomes. They are the autosomes. And these go from pair 1 to pair 22 because there's 44 autosomes. But we notice on the second row on the right hand side there's 2 that don't form a pair. On the right hand side on the second row on the far right there's a very small chromosome. That's actually the Y chromosome. And to the left of that there's an X chromosome. And these are the sex chromosomes or the gametosomes. So we notice that this individual here has 44 autosomes plus 1 X and 1 Y sex chromosome, meaning that they are male. If they were female, they would not have had a Y chromosome, they would have had 2 X chromosomes. So all humans have 44 autosomes arranged in 22 pairs, and then they have a further pair of sex chromosomes, XY for male, XX for female. So in total, humans have 46 chromosomes arranged in 23 pairs, 44 of the 46 chromosomes being autosomes, 2 of the 46 chromosomes being gametosomes. It was back in 2003 that an international collaboration published the genetic makeup of the entire human genome. Now, a genome is the totality of the DNA an organism possesses. And all the cells of the body, as we mentioned before, with the exception of red blood cells, contain a complete genome. They have a complete copy of all the genes. So a kidney cell will contain the information to make a brain cell, and a brain cell will contain the information to make a kidney cell. As you probably know, the genes are composed of DNA, arranged as a sequence of chemical bases. And these chemical bases encode genetic instructions required for the synthesis of protein and for how these protein generating genes are regulated, how they are switched off and how they are switched on. And these bases, the chemical bases, are called adenine, thymine, cytosine and guanine. And it's the order in which the bases are arranged that determines the nature and function of the genes, and therefore it is the order of these bases that determine the proteins that the genes will produce. And bases are often used, or the illustration is often given, that the bases are analogous to letters which spell out the instructions for words. But in this case, the bases, the letters are not spelling out the instructions for words, they are spelling out the instructions to make proteins which make individuals. Now, the way that the order of bases determines the order of amino acids in a protein is that 3 bases will code for one of the 20 basic amino acids that comprise human proteins. So 3 bases will code for an amino acid, then the next 3 bases will code for another amino acid, and the next 3 bases will code for another amino acid. So you build up a chain of amino acids, eventually constructing a protein with all the amino acids in the right order. So physiologically, the genome generates what is now described as the proteome. And the proteome is the totality of the proteins which comprise the body. So genes code for proteins, proteins comprise the cellular organelles and make up the cells, which in turn make up the tissues and organs of the body. Now, I remember teaching this, must have been in the early noughties, sort of between 2005 to 2010. And at that time, we thought there were at least 100,000 coding genes required to make an individual. So 100,000 genes would code for the proteins of the body. But what's been a major surprise, and what I certainly can't currently explain very accurately at all, is that there's only about 22,000 coding genes actually. So in you, there's only about 22,000 coding genes, genes which directly code for making proteins. The rest of the DNA appears to be primarily regulatory. So even though only a tiny fraction of the genome is responsible for the protein coding, it seems that it takes a huge amount, hundreds and hundreds of times more DNA to regulate the activity of these 22,000 genes, saying when it's time to turn it on, saying when it's time to turn it off. They have to be precisely regulated. Now, there's probably an awful lot of other functions for what is called the non-coding genome. That is the DNA in the genome which doesn't directly code for proteins. So the DNA which directly codes for proteins we would call coding genes or coding DNA. And the DNA which doesn't directly code for proteins we would call non-coding DNA. And there's probably, who knows, tens, hundreds of functions of this non-coding DNA waiting to be discovered. And maybe in your career, many of these functions will be subsequently discovered. Well, when he was on the Isle of Patmos, the Apostle John wrote about the Book of Life. And this term has been used by modern biology to describe the genome as the book that describes life. The degree to which that is true can be argued about, and I'm sure it wasn't the book that the Apostle John had in mind, but the term is interesting, the Book of Life. I actually took this photograph at the Wellcome Museum in London, more or less opposite Euston Station. And in the Wellcome Museum, they actually have this biological book of life, where the entire 3.2 or so letters of the entire human genome have been printed out. And each of these books with the numbers on relates to the particular chromosomes. So we can see book number 7, or the 3 books of number 7, comprise the letters in the 7th chromosome. And we can see that the 8th chromosome covers 1, 2, 3, 4, 5, 6 books in the to describe the letters or to list the letters of the 8th chromosome. So each of these chromosomes could be considered as chapters in this whole book of life, describing the entire human genome. So when you get a bit closer, you can see that the book consists of just a sequence of the 4 letters. The 4 letters that represent the 4 bases: adenine, thymine, guanine, and cytosine. So the entire book basically is 3.2 billion letters, A, T, C, and G, in the order in which they occur in the human genome. When you actually look at a page in detail, you see it's absolutely full of the very small print of the correct order of all these bases. And because of this ordering, from molecules to the whole body, some have suggested that a comprehensive, complete understanding of the genome and proteome will provide a total understanding of the molecular basis of health and disease. It's kind of a reductionist philosophy that everything can be explained if only we understood the sequencing of these letters fully. But others argue that the body is so complex it may work as a sort of chaotic system, rather like the weather, a chaotic system. Now, this doesn't mean the body is in any way chaotic, of course, but what it means is that there can be emergent behaviours and properties that are generated by the interaction of billions of molecules. And the behaviour of molecules in chaotic systems is not directly predictable. Therefore, the outcomes may not be directly predictable either. So, basically 2 points of view on that. As ever, the truth probably lies somewhere between those two arguments. Some genes are described as dominant, and a dominant gene is one which will be expressed in the individual, that is, in the phenotype of the individual, if the dominant gene is present. Conversely, a recessive gene will only be fully expressed in the absence of a dominant gene. So in a recessive disorder is caused by recessive genes, and because a recessive gene will only be expressed in the absence of a dominant gene, autosomal recessive disorders are only fully expressed in the homozygous state. That is where two of the genes present in two homologous chromosomes code for a recessive characteristic. And autosomal simply means that the gene is present on one of the 44 autosomes as opposed to one of the two sex chromosomes, that is, as opposed to one of the two gametosomes. So autosomal recessive traits and autosomal recessive disorders are carried by recessive genes on autosomes. But the condition is only fully expressed when there is no dominant gene present, when there are two recessive genes present in the homozygous condition. Sickle cell disease causes a form of hemolytic anemia and is associated with numerous other problems as well. So we normally refer to it as sickle cell disease as opposed to the more old-fashioned term, which was sickle cell anemia. And in this autosomal recessive condition of sickle cell disease, there is an abnormal form of hemoglobin called hemoglobin S present in the red cells. And when this abnormal hemoglobin deoxygenates, it transforms the erythrocyte into this abnormal sickle shape. Now, initially, the sickle-shaped cell returns to being a biconcave disc when the cell is reoxygenated. But after a few cycles, they become rigid and fixed in this abnormal sickle shape. It's also inflexible, and it is the presence of these abnormally shaped, fairly rigid sickle cells that leads to the clinical features of sickle cell disease. In normal physiology, red cells are readily able to deform and squash down in order to fit through the millions of small capillaries that perfuse body tissues. But these rigid sickle cells are unable to pass through small capillaries because they're inflexible and can't squash down to fit through the microscopic capillaries. And sickle cells are also more adherent to the vascular endothelial lining of small blood vessels. And this increases the development of occlusive complications. And these abnormalities result in obstruction of the microcirculation through organs, leading in turn to loss of organ function with acute painful swelling. Plugging of small vessels in the bone marrow also leads to acute, very severe pain in some bones. As the abnormal sickle cells pass through the spleen, they're destroyed at an increased rate by macrophages, resulting in an increased hemolysis and a possible subsequent hemolytic anemia. So the abnormal shape and rigidity of these sickle cells really can result in a variety of manifestations of sickle cell disease. Another autosomal recessive disease is cystic fibrosis, or mucoviscidosis, to give it its correct title. And because it's genetic, the etiology is, of course, entirely genetic. There's not an environmental component in the etiology of this condition. It's autosomal recessive, caused by a defective gene on chromosome 7. Now, the disease presents when a child or young person is homozygous. That is, when they have 2 copies of the defective abnormal CF, cystic fibrosis, gene. And defective genes cannot produce normal proteins. Therefore, the protein which the cystic fibrosis gene codes for is an abnormal pathological protein and does not function physiologically as it's supposed to. And the abnormal protein in cystic fibrosis has been identified. It's called cystic fibrosis transmembrane regulator protein. And interestingly, cystic fibrosis is a disease of white populations. It's very rare in black populations and almost unheard of in Asians. And in white populations, it's about 1 in 25 people carry a single copy of the defective gene. That is, about 1 in 25 people are heterozygous for the condition. Now, these heterozygous carriers don't suffer from the disease. They won't know anything is wrong. But if it so happens that the 1 in 25 marries another one of the 1 in 25, both of the partners carrying one of the defective genes in the heterozygous state, then that can give rise to a child who is homozygous and will suffer from the condition. And one of the main things that happen is the secretions become thick and tenacious. So the mucus produced by the lungs is thick and tenacious and does not clear properly in the mucociliary clearance system. And that gives rise to progressive damage of the lungs. These children are often born with perfectly healthy lungs, although very severe cases may not be. But often the lungs are healthy in early life, but because of the thick secretions and the repeated infections caused by the thick secretions, there's the development of these diffuse interstitial changes getting worse, causing progressive damage to the lungs. So even although these children can be born with normal lungs, the viscous secretions are not effectively transported by the cilia. There's reduced mucociliary clearance. The sputum is viscid and difficult to cough up. It is thick and tenacious. And this leads to stasis of the sputum. It's a general principle in pathology that stasis leads to infection, and this certainly happens in cystic fibrosis. The stasis of the mucus leads to infection. And these patients often have repeated infection. Over time, the lungs deteriorate and bronchiectasis develops, leading to further infection. Eventually, pulmonary heart disease and respiratory failure will develop. And overall, respiratory complications account for most of the morbidity and mortality associated with cystic fibrosis. So the treatment is aimed very much at physiotherapy to promote clearance of the viscous secretions as much as possible, and giving antibiotics at an early stage to prevent the chest infections causing further accumulative damage to these patients' lungs. Now, these boys both suffer from another autosomal recessive disorder called albinism. They are albinos, and albinos do not produce the dark pigments that colour the skin, the eyes, and the hair. So they have very white skin, very often pale, light-coloured eyes, and light-coloured hair. Now, this boy was photographed in the same albino school as the previous two children that we saw, but you can see he's got little patches of pigmentation. But what do you notice about this boy's nose and lips? Do they look European features or do they look African features? Well, to answer the previous question, these children that we've seen on the previous two slides were both genetically African. Again, here we can see one of the children suffering from albinism with some of his siblings. So even although this person is genetically supposed to have dark skin, the albinism prevents the production of the melanin pigments which should make the skin dark. And this, of course, makes the children very sensitive to sunburn, to bright light, and they often have to wear hats and stay indoors a lot of the time because they don't have the melanin that should protect them against the ultraviolet radiation in the sunlight. Well, here we see a genetic form of muscular dystrophy, and you can see the scoliosis in the lower spine. The problem was that because of the chest deformity, this young lady got repeated chest infections because she couldn't clear her secretions properly. But again, you can see the reduced muscle formation in the back and limbs. So this genetic musculoskeletal disorder, as we see, caused abnormal development of the thoracic cavity, meaning that the respiratory secretions weren't able to clear normally. And we also see that there's reduced muscular development in the legs as well. We know what an autosome is. An autosome is one of the 44 chromosomes which is not a sex chromosome. And a dominant gene is one which will be expressed if present. So if an individual contains one dominant gene and one recessive gene, it's the dominant gene that will be expressed over the recessive gene. Now here we see a normal looking kidney. You can probably see the ureter. On the outside we see the normal structure of the renal cortex. In the inside we see the renal medulla, and in the inside, very inside of the kidney, we can see the calyces and the renal pelvis. So that all looks healthy and normal. If one parent is suffering from an autosomal dominant disorder, usually there's a 50% chance that any individual child will suffer from the same disorder. And an example of this is polycystic kidney disease. Now, in the previous slide, we saw normal-looking kidney tissues. These kidneys have got polycystic kidney disease. Poly means many, and a cyst is a fluid-filled space. So there are many fluid-filled spaces in the kidney, progressively damaging the healthy tissues of the kidney. These patients are very likely to suffer from extreme hypertension, high blood pressure in the early stages, but as time progresses and more of the normal kidney tissue is destroyed, the patients will develop renal failure. So if one parent has polycystic kidney disease and the other parent does not, the other parent is perfectly healthy, there's still a 50% chance that any children from that marriage will develop polycystic kidney disease. Achondroplasia is autosomal dominant, and basically the condition is skeletal dysplasia, and there are abnormalities in the growth of bone and cartilage and in the remodeling of bone and cartilage, and they can affect the skull, the spine, and to varying degrees the long bones, and you get reduced growth of long bones resulting in dwarfism. This is a picture of Ellie Simmonds. She's a British Paralympian and she suffers from achondroplasia, although of course she doesn't let herself suffer from it very much. Ellie's a swimmer and won 2 gold medals in the 2008 Beijing Paralympics. And I think she also won 2 gold medals at the London 2012 Paralympics for the 400 meters freestyle. In this picture, we can actually see a blood clot. We've got red cells all held together in a clump or a clot by the fibrin mesh. So the yellow network you can see holding the red cells together is the fibrin mesh. This is normal blood clotting. But haemophilia is a genetic X-linked disorder of blood clotting. Now the common form is haemophilia A, and it's an X-linked chromosomal disorder. It's recessive and it affects about 1 in 5,000 males in the UK. A mother who's a carrier of the abnormal gene has got a 50% chance of having an affected male child, and also a further 50% chance of having a daughter who carries the disorder onto the next generation. And famously, Queen Victoria was a carrier for haemophilia, giving rise to some sons who suffered from haemophilia and daughters who passed it on to their sons. The disorder is caused by a mutation of the normal gene which codes for factor 8 on the 10th chromosome. But it's a disorder of the gene which produces factor 8. And factor 8 is one of the 12 essential clotting factors needed for normal blood clotting for the coagulation cascade. Some cases of haemophilia can be traced back through a family for generations, as we mentioned with Queen Victoria, while others seem to mutate spontaneously, giving rise to new cases of haemophilia, but they can then be transmitted on to future generations. Actually, I've just noticed a white cell at the top left-hand side of the picture as well that's also part of the blood clot. But let's think about clinically what we might see in haemophilia. Well, there can be spontaneous bleeding into muscles, into internal organs and into joints, a hemoarthrosis, where there's bleeding into joints and the joints can swell up and become extremely painful. And in fact, if it's not properly managed, there can be permanent damage to joints. Without treatment, bleeding into the brain is probably the most common cause of death. Sort of intracranial hemorrhages can develop. Effective blood clotting in wounds doesn't occur, so they can keep on bleeding for a long time. And bruising develops, contusions develop even with mild trauma. Hematuria can be present as a result of renal bleeding from the kidneys into the urinary tract. There can be obstruction of the respiratory and gastrointestinal lumens as a result of bleeding and blood clotting. Now, disease severity varies quite a lot between individuals, depending on the amount of factor VIII they're still able to synthesize. So some are very severe sufferers and some are milder sufferers. Treatment consists of giving infusions of factor VIII to compensate for the deficiency. Now, unfortunately, the half-life of this clotting factor in the circulation is only about 12 hours, so it might need to be given on a regular basis via infusions to cover surgery. Sometimes in mild cases, the amount of factor VIII can be stimulated by giving synthetic vasopressin. Now, between 1979 and 1985, there was a great tragedy occurred in the treatment of haemophilia. Many patients were given coagulation factors contaminated with HIV and hepatitis C. This problem arose because many blood donations need to be pooled to get enough factor VIII to be an effective treatment measure. So unfortunately, because people donating blood had HIV and hepatitis C, this was tragically contracted by many people with haemophilia. Now, of course, all the blood is tested, so we wouldn't expect any new cases of this healthcare-acquired infection. Duchenne muscular dystrophy is a recessive X-linked form of muscular dystrophy. The disorder is actually caused by a mutation in the dystrophin gene, which codes for a protein called dystrophin, which is an essential part of muscle cell membranes. Now, this condition usually becomes apparent in fairly early childhood, and there's loss of strength and loss of muscle mass that progresses through childhood. And unfortunately, by the time these children are teenagers, they're often confined to a wheelchair. So progressive loss of muscle mass and strength caused by a genetic abnormality carried on the X chromosome. A common X-linked disorder is blue-green colour blindness, where the individual finds it difficult to differentiate between blues and greens. Now, this does occur in girls occasionally if they're homozygous, but much more typically it occurs in boys. So, have a look at all the numbers in all the circles and see if you can see them all. If you can, it means you're able to differentiate between blues and greens effectively. Now, these conditions we've just been looking at, these genetic disorders, have been primarily affecting a single gene located on a single chromosome. But there can also be disorders which can be included in the genetic spectrum of disorders, which affect whole chromosomes or part of a chromosome. So sometimes there can be an extra chromosome, making 47. Sometimes there might not be enough chromosomes, meaning an individual only has 45 chromosomes. Other times there can be a translocation disorder, where part of a chromosome becomes abnormally grafted onto another chromosome. So chromosomal disorders affecting whole chromosomes as opposed to individual genes. The classic example of a chromosomal disorder is Down's syndrome. Down syndrome is classically a trisomy 21 syndrome. There is an extra 21st chromosome, meaning instead of having 2 chromosome number 21s, there are 3 chromosome number 21s, giving rise to a total of 47 chromosomes. Other cases of Downs are actually caused by a translocation disorder, where there's additional 21st chromosomal material grafted onto an additional or another chromosome. So it means either way there's too much chromosomal 21 material, giving rise to the numerous features of Down's syndrome. Turner syndrome is caused by deficiency of one of the X chromosomes. So these individuals will have 44 autosomes and 1 X chromosome as opposed to 2 X chromosomes. And of course they are all female. And the vast majority of adult women with Turner syndrome exhibit short stature and infertility.
Hypercholesterolemia is the most common single gene disorder causing abnormal lipid metabolism. And in the heterozygous form, it affects about 1 in 500 people in the UK. Now, heterozygous means that one of the genes on one of the homologous chromosomes is normal, but the other gene on the other homologous chromosome codes for this disease, hypercholesterolemia. And this is an autosomal dominant condition. This means if one parent has the condition, there's a 50% chance that any children are going to be affected. And also it means if you come across one sibling who has the condition, we should be thinking very seriously about testing their brothers and sisters, because if they're sharing 2 parents, one of whom has hypercholesterolemia, then there's a 50% chance that any siblings of an affected individual are also going to suffer from the disease. And if we find cholesterols of 9 to 13 millimoles per litre, this is a good indication that this person is carrying 1 copy of the hypercholesterolemia gene. And as we say, this affects about 1 in 500 individuals. Now, very occasionally, we do come across someone who's homozygous for this gene. Thankfully, it's very rare. But someone who's homozygous for the gene, they could be the offspring of 2 heterozygous parents. And people that are homozygous for the hypercholesterolemia gene will present with cholesterols of about 18 to 31 millimoles of cholesterol, total cholesterol, per litre of blood. And these people have refractory atheroma from childhood and die at a tragically young age from the complications of atherosclerosis. They become very, very arteriopathic at a very young age. Well, of course, there's no point knowing about these conditions if we don't know how to treat them and how to advise the patients. Well, if there's a homozygous hypercholesterolemia, these patients don't respond well to any treatment. In fact, the only treatment that's been demonstrated to be effective in the homozygous form is liver transplantation, which of course is quite a drastic treatment. But for the heterozygous form, we advise all of the usual sort of treatments that you would expect. So these patients will need to be on high doses of statins or other drugs which lower cholesterol levels. And of course, we particularly want to lower the low density lipoproteins. So if you can think of any way of raising their high density lipoproteins, that's going to be protective for them, in addition to lowering the harmful low density lipoproteins. Now, diet is of relatively limited value, but we should advise it that these patients should take low amounts of saturated and preferably no trans fats in their diets, and they should replace these fats with things like fish oils, nuts, and olive oils. And also in terms of diet, we would want to give them plenty of fiber, particularly the water-soluble fiber found in fruit, vegetables, and oats, because these can help us to excrete some cholesterol in the stools. Obviously, these patients want to keep as fit as possible and do plenty of exercise. And it's absolutely vital that these patients are not exposed to other risk factors for the development of atherosclerosis. So it's absolutely vital that these patients don't smoke. They should not become obese. They should remain thin. It's essential that their blood pressure is basically as low as possible. We want their blood pressure to be as low as it can be without them actually falling over. And of course, we don't want them to become diabetic. And if they do become diabetic, we would want to treat that assertively. And it's also important to keep an eye on things like thyroid hormone levels, because hypothyroidism is also associated with increased levels of cholesterol in the blood. And if we manage these patients well, we're going to greatly increase their quality and indeed quantity of life. The actual genetic abnormality in hypercholesterolemia affects a gene called the LDLR gene. And this LDLR gene codes for a transmembrane protein receptor found in the cells of the body, particularly in the liver cells. And what this transmembrane receptor protein receives is low density lipoprotein. And it allows for the low density lipoprotein to be taken from the blood into the cells, particularly into the cells of the liver. And of course, if the low density lipoprotein is taken from the blood into the cells, where it can be processed, where the fats can be used to produce energy, if it's taken from the blood into the cells, that means it's no longer in the blood. So it's as if the door is open for the low density lipoprotein to go from the blood into the cells. But if that door is jammed shut and doesn't work, then what that means is all the low density lipoprotein is left standing around in the street. It can't get into the house. So what this means clinically is that the low density lipoprotein cannot get through the defective receptor into the cells. Therefore, the low density lipoprotein is stuck in the blood, and the levels of the low density lipoprotein in the blood are going to increase. And the higher the levels of LDL cholesterol in the blood, the more atherosclerosis is going to develop. The patient will become hypercholesterolemic, particularly because of high levels of the harmful low density lipoprotein. Now, hypercholesterolemia is actually a very complicated disease because you could argue that it's not one disease, it's many hundreds of diseases. And to date, many hundreds of different mutations have been identified in the LDLR gene. And all of these mutations result in absent or diminished receptor function. So many different molecular abnormalities of the gene can result in many different molecular abnormalities of this transmembrane LDL cholesterol transporter and receptor protein. So it malfunctions in many different ways. So the hope here is that individuals, and actually individual families, that their molecular abnormality can be identified and bespoke treatments for their particular molecular abnormality could then be prescribed. Because what you actually find is that each family with hypercholesterolemia often has a unique molecular disorder, a molecular deficit that is unique to that family. So while we can treat them genetically, it would be so much better to treat the underlying molecular basis of the disease. So as we look at our patients, we might notice features which arouse our suspicions. For example, we might notice xanthomas. Xanthomas are fatty, cholesterol-rich deposits in the skin, and they can actually occur just about anywhere, but typically they're in these sites here, the elbows, the knees, the buttocks, and around tendons. And if you feel them, they do feel sort of fatty. Once you've felt a few, you'll get the hang of it. And then a condition that I've probably seen more commonly is xanthalasemas. Xanthalasemas. And these are the fatty deposits around the eyelids. Sometimes you might see one, sometimes they might be multiple deposits. And these need to arouse our suspicions. And then you might notice the arcus senilis, and this is a ring around about the eye. It's sort of an opaque ring around about the corneal margins, and there can be different colours. It can be white, grey or bluish in colour. And also, if patients are obese, we should consider the possibility of hypercholesterolemia as well. So all these are possible clinical features of hypercholesterolemia. We might be wrong, the patient may not have hypercholesterolemia, but it's a relatively easy blood test to check, because all these patients might have very high levels of harmful low density lipoproteins in their blood, which of course will be atherogenic. The human genome contains 3.1 thousand million, that is 3.1 billion base pairs of information. But despite this absolute mass of genetic information, there's probably a little less than 23,000 protein coding genes, giving rise to the human proteome. So this means there's a huge amount of DNA in there, a huge amount of genetic information. And to be quite honest, we're not sure what most of it does. So what's happening now is people with particular conditions are giving DNA samples, and this is being correlated with the conditions that the people suffer from with the exact makeup of their DNA. And this is what we call genome-wide association studies. So we're looking at the whole genome in the context that the genome is all the DNA that belongs to absolutely everyone. And we're using the term genome also as it applies to all the DNA within an individual. So both of these are giving us information. Genome-wide association studies, GWAS, genome-wide association studies. And what these do is allow us to identify areas of the genome that can be associated with common conditions. Now, we may not know how or why, but we can learn that areas of the genome are associated with particular conditions. And we can learn that minor alterations in those areas of the genome can be associated with disease processes. And as we learn more and more and more, we can use this to predict conditions. So it might be possible in the future to take a young person, look at their DNA, and say what diseases they are prone to developing in later life, meaning we can target health education for those individuals. It also helps us to give them a prognosis, and it also helps us to identify relatives who may also be at risk of developing particular conditions. And hopefully, in the medium to more distant future, it will allow us to correct underlying genetic disorders in these individuals. We should no longer be considering our patients as individuals in isolation. We need to find out about their siblings, their parents, their children, and their grandparents, and indeed their great-grandparents or any family history that they have. Because if there's multiple closely related individuals with the same condition, then it's likely that there's a genetic cause. So the more closely related people are, and the more clustering of particular conditions we get, the greater the indication is that this may be having a genetic etiology, which means we can address our efforts in that direction. Now, obviously, we know that there are genetic components in the etiology of a disease, but of course there are many possible environmental components in the etiology of a disease. But with many conditions, the environmental factors have to be present for quite a long period of time before they cause the pathological changes. And with some conditions, if the onset of the condition is at a relatively early age, this means there hasn't been time for the environmental factors to cause the disease process, meaning that the etiology of the disease that we are seeing is probably genetic. And there's some very useful examples here. So, for example, if someone develops breast cancer below the age of 45 or even below the age of 50, we should consider a genetic etiology to that condition. People that develop breast cancer over the age of 50, there still may be a genetic predisposition, but the older someone is, the more likely it is that it's caused by ongoing adverse environmental factors. So let's say breast cancer below the age of 50, we should suspect a genetic diagnosis. And it's the same with colon cancer. If someone develops colon cancer below the age of 45 to 50 years, again, we should suspect that that colon cancer is genetic rather than caused by environmental factors. And it's the same with vision loss. If someone develops vision loss below the age of 50 to 60, again, we should consider the possibility that it's genetic rather than degenerative. And it's also true with dementia. If someone develops what we used to call pre-senile dementia, that is, the onset of the features of dementia occurs before the age of 60, then we need to consider that the cause of that dementia may well be genetic, as opposed to a longer-term degenerative process. So high index of suspicion for these common conditions occurring at younger ages than we would typically expect to see. Now, there's quite a few other times we need a high index of suspicion for genetic components. And one is sudden cardiac death in an individual who seemed healthy. This could be caused by a hyperobstructive cardiomyopathy or a HOCM, which is genetic. Now, unfortunately, this isn't going to help the individual who's already died, but it certainly can help their siblings or their offspring or other family members. Another time to be suspicious is in individuals or couples who have had 3 or more pregnancy losses. that is miscarriages or stillbirths. We need to think if there is a genetic problem here. Or individuals who have multiple congenital abnormalities. Again, the congenital abnormalities are likely to be defective, but if there's several of these, it's worth considering a genetic cause. And another one to mention here is medical problems in the children of parents who are genetically related. So if there's a brother-sister marriage or cousins marry, there's an increased possibility of what we call homozygous load, that recessive genes which might not have been causing problems in the individual come together and can cause problems when they're in the children in the homozygous form. So we want to advise strongly against marrying people that are genetically related. Obviously, we don't want brothers and sisters to reproduce, and we don't want cousins to reproduce. And this is a particular problem in some ethnic groups where they marry within the family. But medically, this is a remarkably bad idea, and we need to educate strongly against this to prevent homozygous pathology in the children who would be the offsprings of such marriages. Now, in exactly the same way that human makeup is determined by human genetics, bacterial makeup and bacterial activity is determined by bacterial genetics. Bacteria are also based on DNA, which is remarkably similar to ours, in fact, essentially the same as ours. And viruses are also based on genetic material. Some viruses are based on DNA, and other viruses are based on RNA. So if we analyze the genetics of individual pathogenic microorganisms, that can give us some very useful information. For example, genomic information can be used to diagnose and treat some infections. Individuals with extensively drug-resistant, XDR, extensively drug-resistant forms of tuberculosis, require rapid diagnosis and aggressive therapy in order to survive. And also, if someone does have extensively drug-resistant tuberculosis, we don't want it spreading to other people either. We want to contain the spread, in fact, contain the epidemic, we could say. And genomic testing of the microorganism identifies the XDR form of tuberculosis more rapidly than other diagnostic procedures. So if you look at the tuberculosis bacteria under the microscope, you can't tell if it's an XDR form or not, but genetic testing can tell us that. And genetic testing can also be used for reliable chlamydia testing, looking at the genome for the chlamydia in a sample. And also, if we take cytology screening, we can look for the presence of HPV, the human papillomavirus, which carries the vast majority, or which causes the vast majority of cases of cervical cancer, because the human papillomavirus is actually sexually transmitted. Well, I'm sure we've all come across so many examples of a drug that seems to work well for one person and doesn't work well for another person, or a drug that is superbly tolerated by one individual, but in another individual patient, they suffer from severe side effects. So some people can take statins, other people can develop a rhabdomyolysis and muscle breakdown in response to taking statins. And it's a well-known observation that people can tolerate alcohol to very different degrees. Like me, you've probably got friends who could go out and drink a dozen pints of beer, whereas I would be sick and would probably die a long, long way before that point was reached. What is it that accounts for the difference? Is it that there are genetic deficits in me or in him that allow different levels of alcohol to be tolerated? Perhaps genetic differences in the production of the dehydrogenase enzymes in the liver that are partially responsible for metabolizing alcohol. There's very different responses to drugs and very different side effect profiles. I've talked to alcoholics who drink huge amounts of alcohol and they say they never get hangovers. Whereas you might not be like that. Maybe you can drink a relatively small amount of alcohol and have a severe hangover the next day because you're not breaking down the toxic waste products of the metabolism of the alcohol. And there are some specific clinical examples as well that we can look at related to drug tolerance and the possibility of side effects, and these being determined genetically. Now, we used to use halothane quite extensively as a hypnotic during anesthesia, but very occasionally when we gave patients halothane, they would develop an extreme, life-threatening hyperthermia. The body temperature would go up and potentially the patient can die as a result of that. And this is genetically predetermined. So if we tested all patients before we gave them halothane, then we would know which ones were going to get this malignant hyperthermic reaction. And of course, we wouldn't give them any halothane. Now, actually, we don't use halothane very commonly now in this country, but it's cheap, so it's extensively used overseas. And it'd be very useful to be able to predict this particular side effect. Or some patients that are given azathioprine, which is an immunosuppressant, they develop bone marrow suppression and can get aplastic anemia and die as a side effect of that. And again, this is genetically determined. Some people are predisposed to bone marrow suppression as a result of azathioprine therapy. Some people are not. And it would be very nice to know in advance if your individual patient was genetically at risk of this or not. And another one I suspect is genetically determined is chloramphenicol. Again, aplastic anemia can be a complication of chloramphenicol. So it would be very nice to know which patients were at risk of bone marrow suppression and potential aplastic anemia and which ones were not, because chloramphenicol is a remarkably useful antibiotic if we could give it safely. An example of how genomic information can be used to predict adverse reactions to drugs can be seen in treating HIV/AIDS patients. Some can develop a fatal hypersensitivity to abacavir. And abacavir is a reverse transcriptase inhibitor. And this hypersensitivity reaction is characterized by a rash, fever, gastrointestinal problems, and respiratory problems. It's a bit like an anaphylactic type of reaction. And we can test patients for a gene variant called HLA-B5701. HLA stands for human leukocyte antigen. So this is a particular tissue type. So we can test patients for this gene variant, and this allows us to predict who will develop this hypersensitivity. And of course, if a patient is genetically prone to developing this hypersensitivity, we won't give them this drug. We'll give them another type of reverse transcriptase inhibitor. Genomics actually has the potential to revolutionize the way that malignant disease is treated. So, for example, if someone's got a tumor in their breast, macroscopically that will just look like a tumor, and they all look the same. But if you do a biopsy of that tumor, you can look at the actual mutations in the genes which have caused the tumor to develop, and you actually find out that there's different genotypes of the malignancy. There's different sorts of cancer with different genetic causes. So, for example, some tumors will have the HER2, human epidermal growth factor receptor type 2. They will express this HER2 receptor. Now, if they do, what that means is it's this receptor that is stimulating the growth of the tumour, because it's a human growth factor. And if there's a lot of this receptor, you'll get a lot of tumour growth. And this is true for 30% of cases of breast cancer. But there's a treatment with what's called a monoclonal antibody for the HER2 receptor. And this MAB, this monoclonal antibody, will block the HER2 receptor. And if it blocks the HER2 receptor, that will block the stimulation of the mitosis, which is increasing the size of the tumour in the breast. So 30% of cases can be treated with Herceptin, but we only know which patients can be treated with Herceptin if we do the genetic analysis for the HER2 receptor. If someone has not got the HER2 receptor, Herceptin will do them no good whatsoever because there's no receptor to block. Another genetic analysis we can do in breast cancer is to look for ER and PR, estrogen receptor and progesterone receptor. Now, if these are positive, if the individual cells are expressing estrogen or progesterone receptors, then it's worthwhile treating them with an anti-estrogen treatment, something that will block the estrogen receptor. We could use a drug such as tamoxifen to do this because it blocks estrogen receptors. And it's the estrogen which can stimulate the mitosis of the malignant cells in the breast. However, if a particular tumour does not have estrogen or progesterone receptors, then there's no point giving them tamoxifen because the tumour growth is not dependent on the estrogen or progesterone receptors. Therefore, there's no point blocking them, and we need to think about another treatment. So what we see is that breast cancer is not one disease. It is not a homogeneous disease. It's many different potential conditions. And we need to do genetic analysis to analyze the makeup of the tumour so we can give the correct treatment for that particular set of mutations that's causing the particular tumour. So one treatment might be absolutely brilliantly effective in one individual and completely useless in another individual. And the only way we can work that out is by doing a molecular and genomic analysis of the individual genetics of the specific tumour the individual patient is suffering from.
Well, here we can clearly see that the nipple is inverted, and this can be a feature of malignancy in the breast. So if in the past the nipple was not inverted and the nipple becomes inverted, that should certainly be assessed to see if there's a possible malignancy in the breast.